can you see CFTR protein?
Yes.
Okay. See it? All right, this is what we're going to talk about for the rest of the period. And I should mention that because you're taking 15 minutes to do a quiz today, the class period will be only 60 minutes rather than 75. What is the CFTR protein? Well, as I said, that the acronym stands for cystic fibrosis transmembrane regulator, transmembrane conductance regulator. The protein is encoded by a gene called CFTR, and it has a specific location on a chromosome. We talked about this a couple of lectures ago, what the chromosomal code means. And here you see the code for CFTR. 7 indicates which chromosome it's on, chromosome number 7. Q indicates the arm. Remember, each chromosome has 2 arms. The short one is called P. The long one is called Q. So CFTR is on the long arm. And it's in a region called region 3. 2 indicates the band within region 3. And 0.1 is the subband. So remember that, what these numbers and letters stand for in a chromosomal code. How big is this gene? Well, it's over 250,000 nucleotides, and that's long, that's a lot. And that all those nucleotides encode 1,480 amino acids. Now, there are many more nucleotide base pairs than required for 1,480 amino acids. Why is that? Well, the explanation is introns. We haven't talked about introns so far, but we'll do that now. On this slide, exons, and an exon is a coding for protein, region, and DNA. The exons are the wide gray bars. The introns are the narrow gray bars. So for CFTR, there are 27 exons and 27 introns. You can see them all displayed out here. So what happens is that the introns are processed. They're removed so that only the exons end up represented in the messenger RNA. And that happens like this. Intron splicing to make a message. If you look to the left, the top line is DNA, which has both exons and introns, and that is transcribed into an RNA that's called a pre-RNA because the exons and introns are both still there. But then the introns are spliced out so that all that's left are the exons, and they're stitched together into one continuous string. And from that are translated the protein, the CFTR protein, first as a primary structure, and then it's eventually folded into a tertiary structure, which is its active form. Okay, why are there introns? That's a big question that geneticists have worried about for a long time. There's still a lot we don't understand about what. But they occur in essentially all eukaryotes. And splicing is done only in the nucleus, as it indicates here in the lower left. But I introduce a new term for you, ribozymes. Ribozymes can be encoded by introns. Now, what is a ribozyme? The bottom center here, it's an additional type of RNA for you to remember. We've already talked about a lot mRNA, the message, tRNA, the amino acid specifier, and rRNA, the ribosomal component that's the machine that makes proteins. Well, ribosomes are none of those, or ribozymes. Ribozymes are catalytic RNA. I'm getting some feedback from somewhere. Let's see if I can. Could you mute your mic, everybody? Thank you. Okay, ribozymes are catalytic RNA. So they're catalytic in that they convert substrate to product, but they're not a protein, they're RNA. And this was a huge surprise when it was first discovered a few years ago. But if you look on the right here, what ribozymes do, one of the things they can do is digest themselves. They catalyze the hydrolysis of their own phosphodiester bonds. At the bottom there, you can see that this digestion is site specific. The ribozyme only cuts itself at designated points, so that the pieces that are left after digestion are all the same length. Ribozymes can be involved in gene regulation. This slide shows how that can happen. There are other ways, but if a ribozyme is encoded near a gene, near the coding sequence of a gene, on the left you see if it's inhibited, the gene can function. But if it's not inhibited, the ribozyme will cleave itself, destroy itself essentially, and the gene isn't expressed. So it's a way of determining whether a gene is expressed or not. Another level of gene regulation.
One more time, what was that? What is that determined again?
In this slide, what's determining it?
Yes.
It says inhibition by small molecule. It doesn't have to be a small molecule, but anything that inhibits the ribozyme will allow the gene to be expressed, because the ribozyme isn't active if it's inhibited. But if there is no inhibition, the ribozyme essentially commits suicide, it cleaves itself, and the gene is no longer functional. So ribozymes can determine whether or not a gene is expressed, let's say at some point in development. Another level of regulation is alternative splicing. So here at the top, you see exons, 3 exons, and 2 introns. If both introns are spliced, all 3 exons are joined, so you get 1, 2, and 3. Or number 2, an exon can be skipped, in which case you end up with a different sequence completely, just exons 1 and 3. There can be alternative splicing. So the illustration here shows that either one end of exon 2 can be there, or the opposite end can be preserved. And sometimes introns are retained in the bottom and become part of the coding sequence. So depending on how introns are spliced can determine the protein, and therefore is a level of gene regulation. All right, getting to the protein itself. The CFTR protein is located on the surface of many different cells in the body. It's a single chain of amino acids. There are 2 transmembrane domains, 2 nucleotide binding domains, And there are 12 transmembrane domains, 6 on either side here. And these transmembrane domains make a channel through the plasma membrane from inside to outside. And that channel is for the passage of chloride ions. So if the channel either isn't there or it doesn't work properly, chloride ions are either transferred not at all or transferred poorly, and things get clogged up on the outside of the cell. Now, there are 2 NBD domains, and I'd just like to take a minute to explain what the nucleotide binding domains are for in this slide. So this transport of chloride ions is an active process. That is, it requires energy in order to get a chloride ion through the membrane. Where does that energy come from? It comes from ATP. ATP is the main energy source in cells. And when you look at ATP and its derivative, ADP on the right, you recognize that that looks very much like a nucleotide. And in fact, it is. Nucleotides are the building blocks for DNA and RNA. But here we're talking about a completely different process. It has nothing to do with genes. It has to do with production of energy. So ATP has 3 phosphate groups at the 5' end. There's an enzyme called ATPase, which can cleave off the terminal phosphate group, leading to ADP plus energy. And that energy is then used to transport the chloride ions. They have to go through the membrane. They get their energy from ATP by virtue of these nucleotide binding domains. Now this is a diagram of what the CFTR protein does in order to get from the gene in the nucleus to the plasma membrane on the outside of the cell. It's a very complicated process. The gene is transcribed, get a message. The message leaves the nucleus to the endoplasmic reticulum, where there are ribosomes. And the primary structure of the protein is made in the ribosomes. It then goes to the Golgi apparatus, where it undergoes a process called glycosylation, which means it's decorated with various different sugars. And from there, it goes through a vesicle, and finally it ends up in the plasma membrane. And if it's a wild type or a normal protein, that's where it functions, transporting chloride ions. Now, this is important to you because your mutant is going to have a problem at one of these stages. And you're going to be able to say, after you study it, what that problem is. And if you remember, one of your slides is going to show how the protein is changed by mutation. And another point that you will make is how its function is changed. Now there's a video here that I don't want to take time. I want to make sure I get through this slide set. If we have time at the end, I will come back and we'll watch this video. But this slide set is in your Canvas container. And I encourage you, if we don't get to it today, to watch this video, because it tells you a lot about this process, how the protein gets from the nucleus to the plasma membrane. I've included several screenshots from that video, mostly focusing on the Golgi and the attachment of sugars to the protein. These sugars are necessary for it to be active. And as I said, that process is called glycosylation. And if we go to the next slide, proteins are glycosylated with both core sugars and complex sugars. Core sugar
What does it mean to be glycosylated?
means sugars are attached. It's really just that simple. So you think of a protein or an enzyme as just a string of amino acids that's folded, but in fact, almost all proteins, in order to be active, have to have sugars attached. And that process is called glycosylation. The sugar that is attached could be what's called a core sugar, which is simply a string of sugar molecules, monomers, linked together. Or they're called complex sugars, which would be things like glycogen and starch, which are highly branched, which you see illustrated on the right here. So glycosylation involves both attachment of core sugars and complex sugars. And now I'd like to get to the really interesting part of this, which is mutations. What is a mutation? It's a change in the nucleotide sequence of DNA. It's that simple. Any change is a mutation. It may result in a change of the amino acid sequence of the protein, or it may not, as we'll see. If a mutation causes a change in the amino acid sequence, the protein may not function correctly. And again, it may or may not function correctly, as we'll see. Mutations are the basis for all variation in living things. Mutations are the raw material for natural selection to choose the fittest to survive. And mutations cause genetic diseases. For example, cystic fibrosis. Cystic fibrosis is a genetic disease. It does not occur without mutations. What causes mutations? Well, you remember that DNA has to be replicated when cells divide. And when it replicates, it's a complicated process. As you know from last lecture, and errors can be made. Any error in copying is a mutation. There are chemicals that damage DNA. And for example, carcinogens. Cancer also is a genetic disease. And chemicals that cause cancer usually also damage DNA. Radiation, go out in the sunlight, can damage DNA, cause mutations. And at the bottom there, I've indicated genome editing. And we'll discuss that in some detail later. And I'll tell you about this new method of making mutations called CRISPR. And CRISPR is an amazing process and an amazing tool, and it really, for the purposes of making mutations, makes all the rest of these obsolete. Genome editing is now the best way to make mutations. And I'll tell you about that at a later time. Okay, how does it actually occur? Well, here is an example of how UV radiation can cause a mutation. Now, before discussing that, look at the lower left, inside the red box. DNA has 4 nitrogenous bases, adenine, guanine, cytosine, and thymine. adenine and guanine are called purines, whereas cytosine and thymine are called pyrimidines. And just to the right there, you'll see the hydrogen bonding between the pairs A's and T's, G's and C's. And above, you can see two thymines on a DNA chain, and they're adjacent to each other. Now, if UV light strikes that DNA, it can cause a bond to form between those two thymines. And that's called either a thymine dimer or a pyrimidine dimer. And to the right, you'll see the consequence. You see the two adjacent thymines, UV light strikes and it causes a fusion between those two thymines. And now that causes a kink in the DNA. So when DNA polymerase 3 comes along, copying, say for cell division, it comes to this kink, it may make a mistake. It may put in the wrong base. It may put in 2 bases instead of 1. It may skip a base. But any change in the DNA sequence is a mutation caused by a kink, caused by a thymine dimer. So let's take a look at the effect of mutations. So this is another example. We went through one of these earlier. But in this case, we're going to talk about the effect of a mutation. So the top in yellow is the DNA sequence of the original strand. And a mutation occurs. So the T in the original strand becomes an A. What is the effect on the peptide? Well, first, you need to transcribe the DNA to a message, to mRNA, and then translate to protein. And so I would like you to do this when you study this slide set. And here is another mutation example where this G in the original strand becomes an A. What is the consequence of that? Again, I would like you to work through that on your own to show that you really understand it. But I've done it for you with a summary on this slide. So we'll look at the 3 boxes, orange boxes, at normal, mutant 1, and mutant 2. Okay, the normal sequence, when you transcribe it and translate it, ends up with a peptide, methionine, serine, asparagine, cysteine, arginine, and stop. That's wild type. Now, in the case of the first mutant, this T in the original strand becomes an A, and if you, when transcribed, becomes a T, but remember RNA doesn't have a T, so it's a U. And so instead of an AGA sequence, we have a UCU sequence. And if we go to our codon table, U is the first base, second is C, and A, they all in this box are serine. So the sequence doesn't change. The original sequence has a serine in this position. The mutant sequence also has a serine in this position, even though there's a mutation in the DNA. So this is called a silent mutation. DNA changes, the peptide does not. Then, if you take a look at mutant 2, this G in the original sequence becomes an A. And when transcribed, that A becomes a U. So now instead of of the UCA codon, we have a UA. Again, if we go to the codon table, U, U, A, first, okay, the first, yeah, it's UUA. First base is U, second base is U. And the third base is A, which is leucine. So now, if you look at the peptide sequence, instead of being methionine serine asparagine, it's methionine leucine asparagine, cysteine, arginine, and stop. So that is called a missense mutation, where you actually have not only a change in the DNA sequence and the RNA sequence, but it results in a change in the amino acid sequence as well. So the point here is these two problems illustrate, first of all, a silent mutation and then a missense mutation. And that leads to definitions. And I'd like you to remember all these different types of mutation, because as you study genes, all of these become important. So a point mutation is a change of one base pair. And we're going to talk about this table in a minute, but all the mutations in this table are point mutations. Some mutations are duplications, which is a piece of DNA is copied, it's repeated, and it's called a duplication. And because it changes the sequence, it is a mutation. A deletion is just that. A piece of DNA is removed. An inversion, a piece of DNA is flipped over so that its sequence runs backward. It's a very large mutation. An insertion can be, is a new piece of DNA is introduced, and an insertion can be anywhere from very small, 1 or 2 nucleotides, to very large. It could be a kilobase or more. And a translocation is usually 2 pieces of DNA just change places with each other. So they can change places on the same chromosome or between 2 different chromosomes. There are translocations that are not reciprocal. That is, just a piece of DNA moves from one place to another. But most translocations are reciprocal. They change places. The effects of mutation on the polypeptide. A silent mutation has no effect. We've just talked about a missense mutation, where one base pair change alters the protein. A frame shift is the addition or loss of base pairs so that the reading frame is changed. And a nonsense mutation, remember this, a nonsense mutation introduces a stop codon. So the peptide terminates prematurely. This diagram to the right here illustrates these things. If there's no mutation, you see the amino acid sequence. There are 3 different base substitutions here. The first one is silent, where the DNA changes, but the peptide doesn't. A missense mutation is a change of 1 amino acid, and a nonsense mutation is the introduction of a stop codon, so the protein terminates prematurely. And then there is a frame shift where you get a completely different DNA sequence, which results in a completely different peptide sequence of amino acids. All right, you have a list of 13 CFTR mutations, and you're going to choose one of those. But that's from among over 2,500 different mutations that are known in this one gene, CFTR. Now, most of the mutations in the CFTR gene are either single base pair substitutions or small deletions. The most common mutation is called F508del. And what that means is that at position, nucleotide position 508, there's a deletion of 3 base pairs, which leads to a loss of the amino acid phenylalanine. That's what the F stands for in the code. This is the most common of the mutations in CFTR. And about 70% of the people who have the disease is caused by this mutation. Many, if not most, of the 2,500 mutations do not result in disease symptoms. Only about 400 of them do. And about 1 in every 31 Americans carries one mutated gene. If you only carry one, you have no symptoms. You have to have 2 mutations in the CFTR gene in order to have symptoms of cystic fibrosis. And I included this just to illustrate visually how this can happen. These 2 blue lines are the 2 chromosomes, chromosome 7, with a mutation on each of them, deletion F508, and it's the same deletion in both. So you got one of these from the father, one from the mother. It's the same mutation in both chromosomes. So that is a deletion F508 homozygote. Doesn't have to be that way. You could have one CFTR mutation on one chromosome and a different mutation on the other chromosome, and you've got cystic fibrosis. Or both mutations could be on the same chromosome. So getting back to your project, your mutation is going to fall into one of these 6 classes. Class 1 occurs in the nucleus, where you get no messenger RNA at all, or no protein at all. Class 2 is not decorated properly. Then there are classes 3, 4, and 5, which affect the translocation of the protein through the cell on its way to the plasma membrane. And then class 6 mutations, you have the protein in the plasma membrane, but it doesn't function properly. Either it doesn't transport chloride ions, or it transports them abnormally, less than optimally, and the cell suffers as a result of not being able to get chloride ions out. And again, this table, your mutation is going to fall somewhere in this table. If you start at the left, a normal protein has no mutations, has no defects. The mRNA is produced, the protein is produced, it's translocated through the cell, ends up in the membrane, and transports chloride ions. Class 1 mutations, you get no functional protein. So there's nothing in the plasma membrane, no chloride transport. Class 2 mutations, the protein is produced, but it's not folded properly, and so it never gets to the plasma membrane to have a chance to function. Class 3 mutations, the protein gets to the plasma membrane, but the chloride channel is defective, and there is no transport of chloride. 4, 5, and 6 mutations, in all cases, there is a protein in the plasma membrane. It's abnormal, and chloride ion transport is abnormal. Okay, good, we're on time. What I'd like to do now is try to go back to that video that I skipped and show you that, just to emphasize the importance of this process going from gene to plasma membrane for your project. Okay, I'm going to stop sharing this screen. You should see a hand, do you?
Yes, we do.
Okay. Then we're going to play it.
Chromosome 7 in the nucleus. So if we have some cell here, so here's our epithelial cell, then in the nucleus, what we have is we have 23 pairs of chromosomes. Now on chromosome 7 is where you will find the CFTR gene. So let's take A, chromosome 7. So what we have here is chromosomes have two arms, and one is a short arm, this top one here. So this is the short arm of the chromosome, and the other is the long arm. Now the short arm is referred to as the P arm, and the long arm is referred to as the Q arm. So this is the P arm, and this is the Q arm. Right, and this is chromosome 7 that we're talking about. Now, somewhere in chromosome 7, you will find the CFTR gene. So let's pull it out and imagine we've got it over here. So we've got this strip of DNA here, which is going to represent our CFTR gene. So I'll just complete the double helix. Okay, so let's imagine that, where's my highlighter here? Let's imagine that this represents the whole CFTR gene. So all of this is the CFTR gene. Now the CFTR gene is a very, very big stretch of DNA. It consists of 250,000 base pairs. So basically, if you count the number of organic bases that are paired in this strip of DNA, and you go all the way along from the start of the CFTR gene to the end of the CFTR gene, you will count around 250,000 of them. So that is a big strip of DNA. Now, the protein is not coded for by a gene that size. Most of it is non-coding. So most of it is what are known as introns. So basically, there are strips of this DNA that are actually going to be used to create the CFTR protein, and those are referred to as introns. So let's draw them on. So basically what we're going to have is little strips of this that are actually used for coding and used to actually make the CFTR protein itself, and all the bits in between are just junk, basically. They're not going to code for anything. So these are known as exons. So between these two lines is an exon, a bit of the DNA which we're actually going to use to construct something. And basically in the whole CFTR gene, in this whole 250,000 base pairs, there are 24 exons. And all the bits in between, so these big bits here, these are introns or non-coding DNA. Right. So when we transcribe this huge, great CFTR gene into mRNA, what we're going to do is we're going to transcribe all 250,000 base pairs, and we'll end up with a strip of mRNA like this. And if I mark on the exons on here again, so here are these 6 exons that I've drawn. Of course, in reality, there'll be 24 of them. But I don't want us to draw 24. So these are the exons. These are the bits we actually want to use to make our CFTR protein. And all the bits in between are introns. So this is an intron, and this is an exon. Right. And this overall is a piece of mRNA. But this is actually what is known as pre-mRNA, because it's the bit that we just literally transcribed from the DNA. before we're actually going to make it, before we're actually going to use it to make protein I, before we're actually going to put it through a ribosome and let translation occur, what has to happen is you have to cut out all of these introns, these bits that aren't going to code, and just stick together the exons, basically, to make what is known as a mature piece of mRNA. And that process is known as splicing. So this is the process of splicing. So basically what we're going to do is we're going to take this intron here, the start of this exon, we're going to get rid of the introns. So this is this first bit of DNA, of RNA that we actually want to keep. Then we're going to take the second bit from here. Then we're going to, so I'll just mark it with this. Then we're going to take the third bit from over there, we'll end up with the fourth bit from here, fifth bit on, sixth bit on. So basically we're going to cut out all the clutter and just keep the coding bits and then join them all together. And then what we get is a mature piece of mRNA. This then goes on to the endoplasmic reticulum, which is a membrane-bound structure which is very closely associated with the nucleus of the cell. So this mRNA is going to go up into this endoplasmic reticulum, and it's going to be translated in this endoplasmic reticulum. So I'll draw this piece of mRNA here. So it's just a strip like that. And basically what is going to happen is that you're going to have a ribosome here, which is going to use this mRNA to make you your CFTR protein. So you end up with your CFTR protein. So I'll draw my CFTR protein here, okay? So this is the CFTR protein. Right, and now what I want to discuss is what is going to happen to the CFTR protein next, basically. So next what happens is that in the endoplasmic reticulum, the CFTR has what is known as core sugars added onto it. So basically what is meant by a core sugar is, so if I do an example down here, if you remember from biochemistry what a glucose molecule looks like, it's basically this hexagon molecule. And you'll remember that if you make starch or glycogen out of glucose, what you effectively do is you bind glucose to glucose over and over again. And you bind the hydroxyl group on this first carbon, because this is actually an oxygen over here. So this is counted as the first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and the sixth carbon actually sticks off of there. Basically, you bind the hydroxyl group of the first carbon to the hydroxyl group of the fourth carbon, and you create these long repeating polymers, basically, structures. And that long repeating structure, if you just continue on like this, that is what is known as a core sugar. It's basically monosaccharides, which is just a single sugar molecule, like glucose, fructose, galactose, bonded together in this predictable manner like this forever. Now, there's another concept that we need, which is a complex carbohydrate or a complex sugar. And basically, complex sugars are when you mess it up, when instead of just doing the linear pattern, so let's do a bit of the linear pattern, but then you also have other sugars bonded onto the different hydroxyl groups. So if we just draw this one here, basically this sixth carbon up here also has a hydroxyl group, so you could make a similar bond like this, but that messes up the pattern and you get much more complicated sugars, and that's what's known as a complex sugar, where you have basically new sugars linked to the old ones of the carbohydrate chain, but you don't link them in a simple repeating pattern from the first hydroxyl group to the fourth hydroxyl group. Instead, you start linking them off of these other chains, basically, and then you get a complex sugar. So basically, in the endoplasmic reticulum, simple core sugars are added to the structure of CFTR. So CFTR at the moment is just pure protein, it's amino acids, but you're going to, it's a more complicated structure than that. So you're going to add on sugars to modify structure, basically. And in the endoplasmic reticulum, the type of sugars that you add on are core sugars. So you add on the core sugars. Then what happens is you traffic the CFTR to what's known as the Golgi apparatus. So here is my best picture of the Golgi apparatus. So the Golgi is a system of interlinked, interlinked membrane-bound structures. And basically what's going to happen is that CFTR is bound in the membrane of this endoplasmic reticulum. The endoplasmic reticulum is going to spit off vesicles, effectively, with the CFTR in. And these vesicles, CFTR, and these vesicles containing the CFTR are going to deliver it to the Golgi apparatus. Okay, so once it gets to the Golgi, so let's draw it in the Golgi now. So let's draw CFTR over here. Once it gets into the Golgi, it undergoes further modification. So you start adding complex sugars to it. So in the endoplasmic reticulum, you add core sugars, and in the Golgi, you add complex carbohydrates. So I drew a simple sugar there, so I'll draw that same simple sugar. So maybe what happens is the Golgi apparatus starts modifying the core carbohydrate so to turn it into a complex carbohydrate. So let's draw some bits coming off like this, which is where you've attached these new sugars basically onto usually the 6' hydroxyl group. Okay, and then finally what happens is the completed CFTR goes into another vesicle and is trafficked to the apical membrane of the cell. And that's all good. So here's the CFTR going off to the apical membrane of the cell. There is also a process for taking it out of the apical membrane of the cell. So once it does get to the apical membrane of the cell, you need another process for removing it from there, should you decide that there is too much of it. And the process is basically clathrin-mediated endocytosis. So here is our CFTR happily up in the membrane now. And basically another process can remove it and return it back into a vesicle, which is clathrin-mediated endocytosis. And these vesicles can then, these endosomes as they're called then, once you've endocytosed something, it's then called an endosome rather than a vesicle. So this endosome can then fuse with a lysosome if you want to destroy the CFTR, so this could go onto a lysosome. Okay, so that's the process by which you traffic CFTR to this apical membrane. So this is clathrin-mediated endocytosis here. So basically what happens in clathrin-mediated endocytosis is that this protein clathrin binds to the undersurface of the CFTR and forms like a net which starts pinching the membrane downwards, and it eventually sort of covers the entire vesicle and just sort of gradually forms an endosome like so. I shouldn't call it a vesicle. These are endosomes. These are vesicles. These are endosomes. The proteins that are in the membrane of an endosome are different to the proteins that are in a vesicle. So endosomes they really did come from the apical membrane. So they have the same sort of proteins in their surface as the cell membrane has. Whereas vesicles have very different proteins in their cell membranes. Well, not the cell membranes, sorry, they're phospholipid bilayers. Okay, so we'll cut this video here, and in the next video what we'll do is we'll discuss how this process is disturbed by a mutation in the CFTR gene.
Are you back to production of normal CFTR protein?
Yes.
You can see that. Okay. I hope that that video was helpful. Again, you can review it on your own time because you have the slide set in your canister, and you will also have the recording of this lecture there. We're running to the end of the period. I said that I'm cutting 15 minutes off because you need to use that for your quiz. Just to give you a preview, next time I want to take a little bit of time to talk about coronavirus. And this is all around us all day, every day. And I'd like you to know a little bit about the details of how coronavirus infects and why it is so particularly infectious. So that is for next time. And for now, are there any questions about anything at all, or comments?
Just to go over, it would be for the quiz today, translation, transcription, and then what else?
Cystic fibrosis, just the part that we talked about on last Wednesday. Remember that it was focused on your project and what's required for your project. And then we went over those documents, which had both information about cystic fibrosis and also a worksheet. You have until midnight, well, you shouldn't wait that long to take the quiz. And then on Wednesday, the first thing we'll do is go over the answers to the quiz.
How many questions?
There are 10 questions.
And you said that you said we were going over cystic fibrosis, the worksheet that you sent us?
Yeah, you should have 5 documents that I sent you, and a worksheet is one of them.
Okay.
The most important one is the one that has all those websites on it. And I pointed out the ones that are essential for your project. The others give you additional information.
Okay.
So for example, if you want to know if there's a drug to treat your mutation, there are websites that will tell you, first of all, if there is a drug, and secondly, what the drug is and what its effects are. So some of those websites are very detailed.
Okay.
Anything else?
Oh, also, when is the cystic fibrosis project due?
December 7th.
Okay.
We will dedicate that entire class period, 75 minutes, to your presentations.
Okay.
Each presentation will have to be strictly timed so that we have enough time for all of you in one class period.
Okay.
That's something we'll talk more about as the semester progresses.
Okay.
All right. Anything else from any of you? If not, I'll stop sharing and I'll see you again on Wednesday. And I hope you've all remembered to sign the chat before I end the meeting. Anyone who hasn't signed the chat, do it right now. Okay. Take the quiz.
Also, is Scott having a workshop today?
Yeah, at 2 o'clock.
Okay, gotcha. Thank you.
Mm-hmm. All right, I'm going to end and see you on Wednesday.